Perfect.
All right. So I just want to start by first to thank Mike for really great lecture yesterday. So I myself worked a little bit in the transcriptomic data, but I think yesterday really he gave a very global view of what different type of modality of data that can be involved and what's their role, and it's really very informative. And you also heard that he's a major player in this large scale BICCN. Mapping the mouse brain. So I think he really brings a lot of perspective to what is the state of the art, what is the challenge of the current one. So we very much look forward to the second lecture today. Thanks, Mike.
Thank you, Yusu. So glad to be here, folks. I see my favorite students are still here. That's good. Okay, good. All right. We had way too much material. We still have too much material. But I've modified some of the presentations, and we're going to kind of do the same kind of tour like we did, except they're going to be topical. I'm not going to go into like great algorithmic details, but there'll be a little bit more kind of mathy stuff today. Okay, this didn't seem to change. Oh, there we go. Okay. Yeah, so the first part is about sort of I call network approaches to cell type data. And this is a broad thing. What does it mean, network? You can think about networks in anything like networks in graph theory, like Ford-Fulkerson algorithm, or minimum spanning trees, this kind of thing, right? All the kind of combinatorial algorithms. And such things have been approached to analyzing data, particularly neuroscience data. I just want to highlight a couple methods here, which these are not what you call deep machine learning methods, but they have been widely used. They're strongly bioinformatics kind of methods that have been used widely. One is this package that was called weighted gene co-expression network analysis, or WGCNA. It was kind of pioneered by these two people here, Peter Langfelder and Steve Horvath. Steve Horvath was at UCLA for many years. I think now he's working for a private research kind of thing. A really smart guy, and he built this out. I'll tell you just a little bit about that. And then there was a colleague who was a student of his, actually, this gentleman, Bin Zhang, who now is I think he's at the Mount Sinai. Yeah, he's in Mount Sinai. But he and a student built another generalization of it too. I'm not going to dwell on this too much, but talk a little bit briefly about it because it's been used so much, and it can apply. Some of these methods may apply to kind of some of the problems that maybe you would look at. So the idea behind this WGCNA is, you can sort of see this here. It's not too, I'll just sort of describe intuitively what you're doing. Is that essentially what you do is you have your objects of interest. In this case, it might be genes for us, right? Or it could be even different parts of the brain or something, anything, could be anything, right? And these are nodes in this graph. And then you make a sort of a set of relationships between them by measuring some distance between them. In case for genes, it might be you might measure the gene's expression across a variety of anatomical areas, let's say, and then you measure a distance between two genes by some simple measure, some correlation. What's the correlation between those vectors, or what is it, mutual information, or something like that, right? And so you have, right now, you just have this complete graph where the weight is the correlation or some other information related thing. And so that's actually, that's what do you do with that? It's pretty general, but there are some things you can do now, but think of this a weighted adjacency matrix now. And I can do things like I can start to find ways to, sort of reduce the dimension of that. I can raise it to various powers. By raising it to powers, essentially, I end up truncating the lower values. I can do sort of kind of community clustering, various things like that. And so there are modules that do this in this. And what you do is that you essentially reduce a graph to the most significant kind of relations between the edges by, these kind of operations, right? And then you build these kind of, essentially you take basically, now you take kind of, you find the eigenvectors of these kind of matrices, and these are kind of principal directions which represent the modes of expression in this graph. Again, it's all pretty basic, but what made this so powerful is that he and Peter, they built like an enormous kind of software kind of repository for all this. And so it's all it's in R. and it's all kind of something that you can, use readily. There's tutorials and stuff. And it works well. It finds interesting kind of things. We've used this a lot, and people for a number of years use this in many contexts. Like we used it to, when we studied our human, I don't have that slide here, but we studied the co-expression patterns in the human brain. So we had vectors representing expression across all these different areas. And in a paper in 2012, when we profiled these 6 different human brains, we use that to find the principal modes of expression across the cortex and stuff like that. Okay, so it's pretty cool. So this gene co-expression analysis has been used, in a variety of ways of kind of functionally kind of determine things. Again, it's pretty basic. It represents kind of coherent kind of, functional kind of, yeah. Graph matching.
The matrix.
It could be, it's, I guess it's, yeah, I guess there's a relationship, I guess, but it's not explicitly a Laplacian. It's actually this adjacency matrix, which has been kind of taken to powers to reduce, the less significant edges. And then there's a kind of, essentially some operations that do cuts, but it's not a Laplacian per se, exactly. But I suppose there's probably a relationship, but it's not, it eludes me at the moment specifically, but what it is. But, and the principle behind this is kind of a bit of sort of what they call guilt by association. If 2 genes are involved, they have so common co-expression patterns, then we imagine they're implicated in some kind of similar kind of a process. Let's see. Yeah. So here's just an example of a GCNA kind of application, and these are the major kind of steps in it here. You're essentially kind of you're forming this kind of a dendrogram using kind of hierarchical clustering, and there's a kind of a dynamic cutting of this, what they call a dynamic cutting algorithm to cut the tree at a various level. And then you end up kind of getting these kind of different modules, and the modules can be interpreted and things like that. So, and then his student they took this and they kind of generalized the method, made it a little bit more kind of powerful, and that some of the ways in which they improved it were essentially, instead of starting with this general graph where everything could be connected, they used an algorithm to something called constructing this fast planar filtered network. And basically what you're doing there is that you're essentially constraining the network to be aligned, embeddable on a sphere. And that means that it has to have genus 0. And that means that you're not going to have, the graph will be planar, okay? And so that by restricting it to be planar, you're reducing the complexity of the local kind of interactions that can occur. So, for example, maybe if you've studied graph theory, people have studied some graph theory? No, yes, some graph, yeah. So in graph theory, there's a beautiful, beautiful theorem dating from like 1930. Right, by someone named Kuratowski, right? Maybe you can know Kuratowski's theorem is a conditions for a graph to be planar. And so the question is, what does it mean to be planar? It means I can draw the graph in the plane without edges crossing. Okay, and I can take like a graph, 1, 2, 3, 4, and kind of draw it like this, and the edges cross, but I can move the edge around so that they don't cross. Well, it turns out that there's conditions, explicit conditions for whether a graph can be drawn in the plane, and it is true if and only if it doesn't contain a complete graph on 5 vertices where everything is connected, and a complete graph with 3 on 1, a complete bipartite graph, 3 and 3. You can think of bipartite graph as 3 and 3, everyone's connected only across, and a K5 where everyone's connected. It's a beautiful, beautiful theorem that you can draw a graph in the plane if and only if it does not contain one of these as a subgraph, okay? And so in this kind of algorithm here, what they do is that they find, using this fast planar filtered thing, they embed this network on a sphere, and this sort of reduces the kind of complexity of the local interactions. And then there's some other kind of, more filtering operations and other things they do to kind of kind of improve the representation. And then you can get better understanding of the modular structures. And yeah, the weights would come in a similar way, either by correlation of you between vectors or some information theoretic thing. That would be the similar.
The embedding, it doesn't change the length.
No, it's topological. Yeah, it's only what's connected. It's not geometrically. Yeah. You're not choosing, for example, like in a multi-dimensional scaling or a PCA, you're not choosing specifically to kind of do that. Yeah.
These methods, they have an n by n matrix.
Mm.
Do they need to keep the whole matrix
Yeah, you would need. I mean, I suppose you could devise ways that you wouldn't have to, but typically the applications have not been of a size where or the order of tens of thousands of variables. It was still okay. It wouldn't be. Yeah. Yeah. What's the what?
What's the difference between the surface of the sphere and then when you do
Well, there's no real difference topologically, but it was just that somehow the ease of the representation, I guess, there was no specific. It could be, you can think of it as being a plane, really. It's not. Yeah, just genus 0 doesn't really matter that much. That's what they do.
It's different logical space.
Yeah. So this is this kind of thing. You can look it up and see if you're interested in this. You can look up these planar maximally filtered graphs. I'm not going to go into too many details. They've been studied. They've been applied in financial data and things like that. The idea is that you get a kind of it sort of increases the local kind of connectivity with 3 and 4 cliques, but kind of reduces overall kind of the planar constraint reduces kind of an overall sort of exponential kind of expansion of the connectivity. And that increases like more small kind of world kind of connectivity and things like that. So, one of the things that has been applied to is, for example, neuropeptides, right? Neuropeptides are important. I'm going to go back and forth a little bit between some biology and more quantitative stuff here, but, neuropeptides are small proteins that act in a more kind of diffusive kind of communication kind of way. They're with neurotransmitters. They're among the most important kind of molecules in kind of in the nervous system. They're larger than neurotransmitters and more slowly acting. They don't act in terms of action potentials, but act in terms of a more diffusive kind of communication. A lot of interest has been in neuropeptides more and more recently. They're evolutionarily kind of ancient, likely existed in nervous systems prior to even kind of neuronal communication. They go back to the Mesozoan. And so they're very important in very primitive animals kind of have these kind of things. So here's a list of kind of some of these neuropeptides that some of them are really well-known genes in the brain, such as neuropeptide Y, somatostatin, vasoactive intestinal protein, CCK, others, and things like that. These are kind of really kind of widely expressing sort of things. And so you can look at these. We can sort of take these. This is sort of looking here at the expression of those group of neuropeptides that we just saw across 285 cell types from the whole mouse brain. Okay, there's kind of and there's pattern, and then you would like to simplify this, right? So we can use these kind of methods to kind of simplify this kind of representation. And so we'll choose to use this more modern, updated package, this Megana. And so you can get this. It's available as an R library. Probably people use R or Python pretty routinely, but if you're going to be a data scientist, you probably want to be able to go interchangeably between those pretty well. So this library, you can get it, you can apply it and download it. And so here I've run it on these, and this is picked out these more kind of the most important kind of modes. It's found these cell types in there which exhibit, they're kind of, in some sense, the kind of the eigenmodes of that expression, right? Kind of pulled them out. And here I've only plotted the output, the eigenmodes of doing this kind of dimension reduction. And on the right, they're just sort of plotted a little bit more kind of the right shows a plot of a set of the neuropeptides in a given cell type. And you know, you can visualize these. There's kind of tools in the toolkit to produce graphical representations and show the connectivity and which cell type. So here, what we're looking at is that we're looking at for the set of neuropeptides, we're looking at what are the distinct patterns and their connectivity that these neuropeptides produce over the set of cell types. Okay, so that's what's going on. These different modules are the primary kind of connectivities and relationships between cell types that occur in that data set by looking at the expression of these neuropeptides across the cell types. Right? And then I can do things like a very powerful, what typically in bioinformatics, people will do things like we'll take this data and we try to understand the annotation of it and what does it mean, right? And so this is a tool developed by a friend of mine, colleague of mine from the Cincinnati Children's Hospital, a big kind of well-known children's national hospital. And this is called Top Gene, right? And typically, I don't know, have you ever used it? Maybe have you used this kind of thing or seen it before? No? Yeah. Anybody seen this before? No? So it's a very powerful tool if you want to analyze. So what's it used for? It's basically used for, you have a set of genes in mouse and human, some organism, you essentially, you put these genes into this, you load them up, and here I'm loading up, what am I doing is I'm loading up the neuropeptides that have come from one of the modules that we found from our previous kind of analysis, right? So on a module by module basis, I might want to know, what are those genes that were in that module? What are they doing, right? And so what does this thing do? How does it work? Well, what it does is that, so there's something called the gene ontology. Gene ontology is this big ontology that's constructed from literature, from people who they've looked at genes and literature has been combed computationally, informatically, and by human annotation, and they have identified different biological processes, molecular function, cell type things that are involved with those genes, and they've annotated them that way. And so if you can think of this, that for every collection of genes, there's going to be some set of molecular processes and things that are associated with it. And then you basically are just doing a kind of like a hypergeometric test, statistical test that says how overrepresented. If I give you a set of genes and I have a given term in the Go ontology, how overrepresented is that set of genes? For example, it might be that, say, what's something here, like neuropeptide receptor binding. So we know from studying all kinds of studies what genes are involved, to the best of our knowledge, in neuropeptide receptor binding. But now I come up with a new set of genes and I say, well, what does this relate to? And so you're doing a test, just basically, kind of a multivariate kind of binomial test, a hypergeometric test that says how overrepresented, how do my genes I have overlap with the genes that are defining those sets? And then it gives all those outputs with p-values and corrected p-values, et cetera, for all these different things. So we can see and interpret it. And we see that, this module here, quite unsurprising. Oh, sorry. Yeah. Sorry. Let me do the cursor. This module here, thank you. This module here, is sort of showing, this to be expected, the various neuropeptide activities and essentially, the corrected p-values of how overrepresented those are. So people in bioinformatics use this kind of thing all the time. Furthermore, you can set over here, a background, like a statistical background. So, you may not just want to, I may have an experiment, but I want to know, conditioned on a set of genes, how overrepresented are the ones that I have, right? And so you can put a background set in, and do similar tests. So that kind of thing is very powerful. So, if you have sets of genes and different things, you can do these module analysis, these network things, and proceed, So such as things like, autoencoders have been a very powerful way of doing dimension reduction. People use them all the time in different contexts. So here just is showing that here we had, remember we talked about yesterday about that we often, and we want to analyze gene structure, we take highly variable or highly differentially expressed genes, right? The genome is big, but a subset of them, those that exhibit differential patterns, are the ones that are often the most interesting. And so on the left, I'm just showing here, kind of what I'm showing here is an autoencoder embedding using 6,083 of the more highly variable genes. One could have just taken those, done some other form of dimension reduction like PCA or something, put it into a UMAP or something like that. I could have seen the visualization. This is just using a kind of, an autoencoder here to reduce the patterning and kind of give another kind of representation. And on the right, what we've done is that we've taken a much smaller set of only using the 47 neuropeptides and to try to see how well, using a similar mechanism, are these actually describing the same kind of space that we saw, right? And so we see it's pretty good. You can see, and you can measure that. There are ways to kind of measure how close we captured the thing. Other genes, if we were to do this with the neurotransmitters, it would probably look even closer, right? But this was part of a paper that we were studying the neuropeptides on, so we wanted to understand how well they align. You can also do things. An individual in my group who's very strong in this kind of thing has developed a lot of other kind of means of using these kind of technologies, and kind of propose these kind of these coupled autoencoders, where here now there's going to be autoencoders working. Remember we talked about multimodal data yesterday. We could be doing this for gene expression data. We could be doing this for electrophysiological data. And we want to learn a common kind of representation between the two, how they associate. And so here I could imagine autoencoders doing them both independently, but I could also couple them in the sense that there's a kind of an interaction term or a penalty term for how well they're agreeing and how well they're trading off. And so that can be modeled, right? I can model this kind of thing by, a classical kind of agreement of essentially, loss in the individual encoders together with a kind of penalization term of interaction, okay? Not an uncommon kind of approach. And this kind of, this has been actually quite powerful in actually seeing and using this to understand how well the electrophysiological data relates to and predicts the gene expression and vice versa, okay? And this is just here showing a kind of, alignment, cross-modal kind of, reconstructions of these, and how well, ultimately the cell types that are predicted from the gene expression data are aligning with those that are predicted. If I try to define cell types by gene expression patterns, and I try to define cell types by, electrophysiology, how well do they agree? And you can see this is just a visual down below, but you can see here that this is a pretty similar looking structure, right? So it's actually this dueling autoencoder thing. It seems to be working pretty well toward capturing a common vision of what the cell types should be. And you can look up that paper. The paper, I don't remember the name of it, but if you look up these guys here, Ugar Sumbul or Rohan Gola, you can see that there was a paper about these kind of autoencoder kind of things. And they've generalized this to multiple kind of things, multiple sets, where now we're going to bring in transcriptomic features, morphological features, et cetera, multimodal, right? And show that it also kind of can work. It's a very good approach to kind of integrating these kind of multimodal data. And this would apply to any number of things. You can think of this, here we're talking about neuroscientific data sets, but you can imagine this working for a variety of data that might be interesting to you. Another problem was this problem of understanding. So this was this problem of understanding, you know, what are the types? And remember how we talked about there were types versus states. We also observed yesterday that there was this problem of whether a cell type was really truly categorically different, or whether there was kind of, you know, gradients between these types. This is kind of a big issue. We don't know. We cluster things. We see kind of, you know, apparently classes that can be resolved by different hierarchical methods, but there's also seemingly a continuous kind of transition between some of these states, particularly in developmental data. You know, as the organism is development, things don't happen in typically big jumps, typically. And so one way to model this is that you can use kind of, you know, distributions where there's an underlying categorical distribution, but that you're able to kind of control the variability of it and to actually model how continuous things go between these kind of categories. Such a thing, a distribution you may have heard of, is called a Gumbel softmax. It's a way of actually modeling a categorical distribution with free boundaries that can move between them. It's a very powerful kind of thing. You can look it up. It's pretty cool. There's some interesting tutorials on that online. But so we've used that actually to study some of these gradients and how categorical versus how continuous transitions between these classes are. On the right here, you see kind of embeddings of this, whereby, you know, using this, we see these kind of really kind of smooth transitions between these different types, and less than, you know, less than really distinct kind of complex. Okay, so this is just basically, this was just kind of basically this introduction to certain network type approaches here in this kind of thing. You know, we talked about you know, more kind of sort of standard graph theoretic ways of, you know, generating matrices, performing cuts, doing kind of basic linear algebra on it, versus kind of more learning approaches here, where we build encoders, we constrain them, we build penalty terms, etc. We use other kind of modeling things. So that'll be all for this little unit here. Do people have questions on this section here? No. Most of these papers I've included in the reading materials, so you can look at these papers, and hopefully this will provide a kind of this provides a kind of introduction to the spirit of what's going on. It should help you to read the papers a little bit more directly. So let's see here now. Yes.
So it's about the same question that I asked before. Is any of these methods for, let's say, million data points?
For what?
Million data points?
Oh, yeah, sure, absolutely, no problem. Yeah, especially these kind of, you know, autoencoder type approaches here would be, that's not a problem. We can do this on big data sets.
Not the graph.
Not so much those. I think that it also, because you'll probably recall, if you're, you know, no doubtly, so raise your hand if you're more familiar with R than Python. It's okay to say, you don't have to be a global expert. I'm not an expert either, okay? You know some R, you've used it a little bit. Raise your hand if you're more familiar with Python. So the Python people have won, okay? But R is good, don't be worried. But the trouble is that R has a lot of nice merits to it. The other thing is that if you're doing big data sets, you know, I think Python is going to be the method of choice, right? It's just due to the way R stores things in memories and the need to kind of package and build this list-oriented kind of structure. It's very memory hogging, right? Although there's ways, you know, you can read these H5 AD files into R2 and do sparse data analysis too, but it's just, I never use R if I'm doing really big data, you know, and I think most people would feel the same. So, but yeah, there's no, I don't believe there is no, you know, a lot of the bioinformatics people started off using R rather than Python, because Python was just less popular in the earlier days. So some of these methods, you know, are not available in Python. The WGCNA doesn't, I don't believe, doesn't have a Those don't have a Python implementation, but I'm sure there's, you know, equivalent kind of things you could do yourself in there. Okay. Okay, so now we're going to move on to, you know, we're going to kind of come in and out of various kind of computational type things. You know, we'll change our orientation from time to time about how biological versus how computational you are. I mean, I know some of you are interested in a little bit more in the biological applications, and some are more interested in kind of the computational details. So we'll try to hit both and stuff like that. So this next segment here, now we're going to do, we're going to do, what we're going to do is a couple things in the rest of the day. We're going to do a deeper look at transcriptomic or omics data analysis and what are the problems and what are the challenges kind of there. And then we're going to, this will be sometimes more biological, sometimes more computational. And then we're going to talk about, in the last part, we'll talk about data integration and using machine learning methods for integrating big data sets and things like that. So, you know, we talked about yesterday, you know, the genome, the transcriptome and a whole stuff. Think about it, it's entirely kind of amazing that we can profile, you know, largely the transcriptomic status of an individual cell at a whole genome level. And there's, you know, I said, yes, we talked yesterday about how that works, kind of, it's basically extraction of material, amplification. Oh, we got a question coming in. Is SQL used in computational? SQL is definitely used in terms of, you know, database manipulation and database kind of organization. It has been classically an important tool in bioinformatics. You know, let's talk just a little about the difference between bioinformatics and computational biology, or in terms of neuroscience, neuroinformatics versus computational neuroscience. What's the difference between all these things? Well, again, there's not a clear boundary between what these are, but I think most people think of bioinformatics as being more the organization, manipulation, and annotation of biological data. And we think of computational biology as being more the using computational methods to derive insight into data analysis, connecting with biological interpretation. And the same, I could repeat those terms for neuroscience, and you have neuroinformatics versus, but those boundaries are not clear. In some sense, neuroinformatics, you could say, is, or whatever it could be, or computational or bioinformatics could be everything. But, you know, these are just the terms that we typically think of. But, you know, there's a lot we talked about yesterday, that what has to happen to profile this. And we're not going to repeat those kind of details, but in the slides here, you'll see that these were the steps often involved in profiling data from a single cell. There's big problems with it, right? The problems that these measurements, they typically suffer from their sparsity, a large fraction of dropouts. What does a dropout mean? It means you're trying to record a gene, but you don't have enough genetic material to determine if it really exists. Or like doublets, things where cells bind together and it's not really two cells, but because the cell was in the process of doing it, they coalesced, and we have an inaccurate representation of what that cell is doing. Those need to be filtered out too. Lots of aspects for noise. There are two major methods that have been used in profiling kind of genomic data. It's worth understanding a little bit about the difference between these. One of these is called SmartSeq, and this was kind of you construct little kind of droplets from your kind of separating the cells, and there's a certain kind of, you know, a process which is used involving kind of barcodes to label them, and it's very precise and very powerful, but also expensive. And it produces a very detailed kind of profile. If you really want to know what's happening in the cell, you really would probably want to use this. This will tell you the true kind of expression of that. Except it's expensive, and you can't get a lot of, you can't get high throughput to process many cells. So the field, in many ways, if you were studying what was really happening in a cell, that you really wanted to understand how it affected people's health or something, or some translational thing, you would probably use the SmartSeq thing. But in the case of like classifying and clustering data and determining the cell types of the brain, you don't need necessarily that level of resolution. You want to cover more cells at a slightly less depth. So what depth here means is that the number of genes I detect in the cell as being expressed. This 10x Genomics company makes, they kind of, through a lot of work pioneered through people like George Church, from Harvard, and they developed these methods whereby essentially you could simplify the method of this to actually kind of get a much higher throughput kind of approach, and you don't get the depth, and you get more dropouts, but we can process many, many more cells. This is just a plot here showing an example here. Here I've taken, I'm showing two different regions here: somatosensory cortex, primary cortex, and visual cortex. And I'm plotting, these are the fraction of expressing cells using the SmartSeq along the x-axis versus using the 10x approach on the y-axis. And so this curve here is essentially just the mean number of cells at every level. And what's being plotted here is just the quantile, essentially the fraction of cells expressing. Okay, so you can see the variability here is quite tremendous. These are putatively profiling the same kind of genes expressing in these regions, but there's a great deal of variability. That sure, it is true that higher in SmartSeq, in other words, more cells expressing in SmartSeq does tend to be higher in 10x, but there's a very nonlinear relationship there. And so this is kind of a problem. So we do a lot of things to kind of understand, using these methods. The 10x method, the lower kind of resolution one, has become a bit of the de facto method of choice for doing large scale classifications, because in the work we've been doing recently, we're more interested in, determining just what are the basic cell types, and do we have enough resolution power to draw a classifier that says I'm separating one group for another. We don't need to know the detailed kind of biology. As we said, there's a lot of different issues here, issues of multiple levels of resolution, where we talked about yesterday how you can't take all this data and put it in one thing and do a big kind of classification, that you really need to try to separate major groups first and then look for finer resolution. And this is done. And you have the problem of, these trajectories of change and gradients, and all this kind of comes in. So these are big problems in single cell analysis. Oftentimes, how do we deal with this sparsity and these drop? Yeah. Yeah. What I mean is, for example, just that. I mean, in the case, take for example in development, right? We could have like an organism is expressing certain genes at some level of the embryo. As it matures, it becomes older. These gene expression changes, and what we call the cell type that we found is sort of at day 10 may have morphed into something else, because it's heading toward a terminal, more adult state. For the adult, it will have changed. And so when we plot this data, we look at this data, we see these kind of gradients and movement. And so there's lots of analysis that goes into this. There are many packages. You can go find these online that have been trying to use these kind of trajectory analysis to see what are the primary modes of how these things are moving. How are these expression patterns changing over time? And so it's trying to understand, really, there's a difference between, so we're really trying to understand the difference between cell state and cell type. What we mean by cell type is something which is unequivocally distinct and will have distinct function ultimately at some terminal state. Cell state will be either due to the differences in the ambience, or distances in development, or something is changing dynamically. And so you really want to be able to model these differences. That's kind of the idea. There's many packages for doing this. And we use something that we use like a concept they call pseudo time. If you've heard it, I know some people have heard of pseudo time. Lucas over there has heard of pseudo time, yeah. And this is the idea that we don't know the actual pseudo time means that we don't know the actual time that is in terms of standard time intervals measured, but we have a way to track trajectory, and we impose on it some order of sequence, right? We make a pseudo kind of time. So there's a lot of work that when you do this data to also imputing values, like oftentimes what we'll do is that you'll have smart-seq data, detailed transcriptomic analysis here. You'll have this coarser 10x. And now I have a new region that I would like to know something about data from that I don't have data for, but I have 10x for, but I don't have the detail. And so I'll try to impute the data using, kind of more standard data imputation things to do that. There's various techniques for that, techniques of data smoothing, data reconstruction, matrix factorization approaches, as I mentioned, and autocoders. This imputing things, here's a list of I'm just giving you kind of a list of things. You can go check some of these out if you want to learn about some of them. There's a whole sort of various approaches to this problem of data imputation and data smoothing. These are kind of a set of lots of different, you can see lots of different approaches from simple, more statistical things, non-negative matrix factorization, all kinds of approaches, different clustering, nearest neighbor approaches. So these are all available through, most of these would be available in our packages. You can check some of those out. Should we do that? So we said enough about that. I don't know, we don't really want to say too much about the difference between these platforms. This information is in the slides. There's another thing too that is important in classifying these genes is the difference between, in the gene, that you have exons maybe and introns, right? Parts of the genes that, is anyone not familiar with exon versus intron? Yeah. Okay, give me the idea. So you have a gene, just give the basic, you have this gene, but what's a gene, right? A gene means it contains material that will be transcribed, which will be made into a protein. But part of what we call the gene, not all of it is made into the protein, but there's a signal in there, which are these things called exons that actually will form the actual kind of messenger RNA, which will be translated. And their spacing in between those are what we call the introns. The introns are not junk, but they oftentimes serve a role in determining, the packaging of it, the kind of regulation of what will be done, the alternative splicing. Like sometimes, it's extremely complicated. What's made a gene is not always the same genetic material, but certain things in certain conditions will be sort of spliced out. Okay, and so, but yeah, I'll get to that in just one second. But so we have, we think of the exonic material as being the protein coding kind of signal, but actually it turns out in terms of classifying the cell types, especially in human, the intronic signal is very important, and certain methods favor discovering what's happening in those introns. Okay, we had a question. Let's see, what was the question? Let's go to the chat. Oops. How do we go to the chat? I got to go to my chat. Yeah, not that chat there. Okay. Let's check the chat. Okay. Given that there are various methods for processing omics data set, and each would have a direct effect on the conclusions drawn, how would you approach just choosing a specific pipeline to make reproducible analysis? Okay, yeah, that's a good question, and that's what we were kind of trying to address. Is that I think that the main, how does a biologist make such a decision? I think your question is how does an experimentalist make such a choice? It will be a factor of often it's a factor of money. How much money do you have to do the job? that, again, I think the big distinction is if your goal was to determine biological function and its implication for an organism, a problem of translation, a problem of disease, a problem of really understanding, you would want to use the higher resolution method like SmartSeq. If your goal is more to simplify, to do dimension reduction in the space of biological alternatives, to classify cell types, to say, I simply observe something different here than something there, but its specific molecular function I'm less concerned with, then you want to use a higher throughput kind of method, right? You can get more data in more cases for cheaper. That kind of, does that help as an answer to that question? That's kind of that. Often that choice is not, the choice is not, you're welcome. The choice is not left to the computational scientist. It's often a choice made by the experimentalist and what they did. But if, for example, you were applying for a grant and you were really claiming to have some method that was going to really offer some benefit to somebody, you would need to be using a higher generation method. And if you were to submit your plan using this 10x, undoubtedly in review, it would come back saying, well, we question whether or not you have the resolution to really make this distinction. You would probably get such a critique. Okay. Okay. So, now that we have the problem, we go to this problem of actually kind of, interpreting the data, reconstructing the data, using machine learning approaches to deducing what we have in the data. And there's all kinds of methods that are used here, from, basic kind of clustering, matrix factorization methods, kind of neural network, variational kind of autoencoders. And in the last segment, we'll talk about some variational approaches to the data integration. But this is kind of just showing you you can check some of these out. You don't have to write these down. They're in the slides. You can look them up and check it out. Oftentimes, one of the big things that a lot of work is going into now, which is not yet, is far from elucidated, is the gene regulation, right? Is it so? We know it's expressed on some level. And we don't want to oversell what we're doing here. A lot of these giant profile things have been done using these 10x methods, for which we see groupings, but we may not have the resolution to really determine fine, detailed resolutions. And we certainly don't know what they do, I think that there's where data like epigenetic data, right? Epigenetic data concerns two different things. One, whether the chromosomal structure is open so that the gene is enabled to be expressing. Methods like ATAC-seq to find that. You can actually measure whether chromatin is open in a single cell using that, or its methylation status, right? The change in a configuration of a nucleotide, which becomes methylated when it's able to be part of a transcription initiation. These are measurable now in single cells, and they're measurable in high throughput. But one of the places where we don't have a lot of insight yet is how these two relate. There's a lot of effort going on using big, complicated methods, AI methods, et cetera, to try to understand how regulation is working with transcription. To do this in a really meaningful way, you would want to capture these data, both the transcriptomic and the epigenomic data, in the same cell. And there are methods that do this. We call these they're called multi-ohm methods. And a lot of people now are using these multi-ohm methods. Again, there are problems of dropout, of statistical significance, and all these things that occur. But, one of the big grand challenges right now in computational molecular biology would be to really understand these relationships of transcriptomic versus epigenomic and gene regulation. The reason is that sure, we know that transcription starts here, but we also know that there are things that affect it, and these can be vastly at some apparent non kind of close relationship in a linear sense, what we call cis-regulated. not close to the transcription start site. It may be that it's affected by something that's kind of transposed kind of off in another chromosome or something like that. So this is a big problem. So this here, this is just a little bit on kind of some practical data stuff here. We recently just finished this. So this was a big output of this Brain Initiative cell census work, was a paper now that was a really large scale transcriptomic and spatial atlas of the whole mouse brain. Okay? It's published in December of last year. And it is, this paper is available together. There was a whole pack in this part of the pack. Remember that there was an original package we talked about yesterday that was that primary motor cortex data, which was the first kind of coordinated deep dive into using different modalities in the primary motor cortex. That took a long time to get just the cooperative groups together for people to do that. And what often happens is that, you do a project and you pick a prototype region and you're going to do it to get off the ground, And typically what people do in this kind of things, they say, okay, we're going to give it a quaint little name, which makes it sound like we're going to bang this out really quickly. So we called it a mini atlas. Whenever you start a project, never call something your mini thing, because you find yourself taking the vast amount of your funding to do the mini atlas. And then you're at the end at 4 years and all you've got to talk about is a mini atlas. Well, so anyway, what happened, but it's actually not it's not really, I'm being facetious. It's not nearly as bad as that, but all the technology and all the cooperative work goes into setting up this kind of working infrastructure. And so now you can actually finish the job in a really effective way. And so, whereas we did spend a lot of time doing the mini atlas, just recently, just following the ending of the BICCN, people released this high resolution transcriptomic and spatial atlas here, which is it's using only the 10x methods. There's no, in earlier versions there was the SmartSeq, but to do the whole brain, it was required to do use this lower resolution method, but it uses this method to profile, hundreds of areas in the brain and the cortex and lower brain, et cetera, things like that. And this is available. All this data is available here. You can go, if you go to the Allen Institute, we built a kind of a resource that that sort of shows this. Remember how yesterday we talked about the difference between the spatial data and the transcriptomic data? Remember that sort of, you have this in single cell transcriptomics, you're basically doing sequence reading and you're mapping them and you're getting gene expression that way, but you don't know anything about the spatial distribution of where those neurons came from, because you took tissue, you disassociated tissue, you ground it up, whatever, and you isolated your cells and then you sequenced them. Well, right, going back to that you really want to know the spatial context of where these genes are active in these cell types. And through these methods of like MERFISH we talked about yesterday, we're able to gain some, understand the spatial distribution. So those data are also in this atlas, okay? Now the spatial thing is, remember, as we said yesterday, you can't do the whole genome using that, but you could get panels of about 500 genes and things. So all that data is available in this atlas. This is just showing here a representative of this taxonomy and how it's organized in a kind of hierarchical way. This is not showing the whole taxonomy, it's just too big, but you can see here it's across different regions. It's what we call the major classes, the major inhibitory and excitatory classes, going down to what we call subclasses and subtypes, blah, blah, and how the different clusters relate to each other from the spatial to the thing. We're talking about how you do this clustering when we organize these atlases, and there are many methods for doing this kind of thing. I'm going to tell you a little bit more about the method we used when we organized this data, but this is just a slide here showing some of the strengths and limitations of various different kind of methods for doing kind of clustering of genomic data here. These were all been developed specifically for omics-based data sets, but they use all kinds of combinations of things, from combinations of standard things like PCA to, k-means based things, and all these things will be familiar to you. They're all kind of using different things. You know, and then we get different representations of this. This is just showing how this is kind of a bit of a kind of a, in a way, a confusion matrix of sorts between the classes. This is an embedding where the dots here are now different cell types. The larger blobs are a higher level organization. And these edges, which can go between these, are a measure of confusion in a way that the arrow is, the line is thicker if upon resampling, we do resampling when we do the clustering to determine how well the clusters hold up. And the line is thicker if there's more crosstalk between the different little things. And I know some of you have seen these kind of pictures before, but it's a way of visualizing what are the major groups and how do they interrelate these.
Also considering this multiple clustering methods.
Right. That's right. Well, that's right.
That's
right. That's right. Another way to approach this problem would be to say, as Yusu was pointing out, to use several different methods, and that has been done. That's been done. And there's methods. There's a nice method from somebody at University of Toronto named Jesse Gillis, who developed a package called MetaNeighbor. And I don't know if we'll have time to talk about it. I have slides about that. It's in the material. I don't know if we'll have time, but the idea there is that what they do is that you have different clusterings from two different things, and you look for these more unequivocal landmarks that are likely to be representative of something. And the MetaNeighbor, essentially using neuroscience approaches, tries to find a match between those and then reconciles what are the consensus types that are in agreement. Yeah. But that's, in fact, that's what's done here. This is based on bootstrapping. That's right. Yeah. Well, okay. Yeah. So here, what's done is here is that, so let's just back up a tiny bit so people don't get, there's nothing worse than people see a picture and you kind of get it, but you don't really get it, right? But let's just hone on this a little bit more. You had data, you had expression patterns of all these different cells. You want to group the cells into groups of which each cell is going to be what you will call a common cell type. Okay. And we compare them by looking at the gene expression across a set of genes, large set of genes, big vector. Think of it as a big vector for every cell. Every cell has a big vector of all the genes expressing in that cell. I have some kind of metric of closeness, whatever, and I do some big clustering effort of any number of methods, whatever we're going to do. Then out of that comes a partition of these cells into groups, which we are calling these are the different points of the types here. And we want to know how robust those groupings that we found are. What's your first name? Yoav? Yeah. So Yoav. Yeah. So Yoav says that we want to know how robust these types are. So what was done here, in fact, was a subsampling on bootstrap sampling of the same data set, and then reapplication, just as he's suggesting, of the same methods, and then repeating this, and then measuring the variability through that exercise. And the lines are measuring how much kind of interchange between, in other words, cells, take a specific example here of a thicker line here and here. Well, these were called different types in the original grouping with the full data, but upon subsampling, we found that cells in this group were often confused with cells in this group. And that's coming out of the bootstrap, right? So this is just representative of this resampling method and how, yeah. So, why that is done, as he's suggesting, is, we don't have, if you write a paper, if you think about it, writing a paper and you're doing a data analysis, you might be tempted to sort of you think, well, I could apply 5 methods to the data, but that doesn't have a sound of satisfaction to it somehow. And somehow statistically, people feel more comfortable where, well, we did this method because we were confident in it, but we want to measure the variability, and we're going to do some kind of bootstrapping or some statistical testing on it. It certainly would be useful to say these results also hold up under this, but it's unlikely that you would, like in the Netflix challenge, declare the winner to be an average of 52 methods that you did to pick movies, right? That wouldn't be very satisfying, I think, in a publication sense. So anyway, that's that. So, there's a clustering strategy that we kind of use. Let me just go. This is kind of the clustering strategy. Look, we're going to take a break for a minute, but I just want to review this kind of thing. This is the major clustering strategy that was used in this determining this whole brain atlas, and the ones that we have used in recent atlases. Okay, so the steps in this is complex flow chart here. Okay, there's going to be a quiz about this flow chart at the end, so I want you to study this. Okay, no. So, but these are the steps here, the major steps in it here. It's pretty straightforward. This is not using any kind of advanced AI methods. It's more standard kind of statistical approach, but there's a lot of formatting and setup involving making sure the data is clean, robust. You have a lot of data cleaning and a preliminary pre-processing is always very important. There's, selection of your hyperparameters and your parameters surrounding, choice of distance, choice of this, what means closeness, et cetera, like that. You need to set those up because you need to sample over some of those. There's this iterative hierarchical clustering, which is done. Then there's, we look for these high variance genes, the ones that are kind of able to determine the difference between groups. Then there's a kind of dimension reduction step, could be PCA, or remember WGCNA, we talked about that right in the beginning. Using the WGCNA to determine the primary modes, right? And then, kind of some filtering steps, and then kind of a clustering on this reduced kind of beta using kind of community detection approaches and looking for modules. And then finally, sort of a cluster merging. Like you find all these groups, but maybe some of them don't hold up. Using the bootstrap, we're going to merge some of them to form a final atlas. And so that's basically an outline of the approach. This is just showing a gigantic UMAP here of the cluster. This is the entire brain here of the mouse on the left, colored by, I'm not sure this would have to have there's like 4,000 clusters, okay, brain wide. So there's, in principle, 4,000 colors here. I don't know exactly how they generated the 4,000 colors. If they're probably some are reused, I can imagine. But in any event, you can see it's a bit messy. Although you still see here that there are evidently regions of these clusters which are really quite individual, right? But when I go up levels of resolution, I more coarse levels of resolution. That as I go up that hierarchical tree, I start to get groupings that become more solid. These are sets of cells now colored by kind of bigger, coarser groupings. And then finally by major classes of, various kind of inhibitory and excitatory patterns that are big groupings in the brain. And you can of course color by neurotransmitters or other kinds of things like that. Don't necessarily, or by brain regions, right? How you could, for example, this could be here a region of, say, genes expressing in the thalamus, okay, over here in this cyan color, right? No, that's right. That's right. It's all data driven, and it actually, interestingly, does separate the brain into, so that you can deduce from this, right, that apparently there's some, it's not a dramatic deduction, but it is an interesting deduction that apparently these regions of the brain have uniquely associated cell types that are somehow doing their own thing, as Yusu is pointing out. And in fact, we did this kind of thing early on, even, is that, so what did we do early on? Early on, we remember talked about yesterday the original mouse atlas, which was this in situ hybridization atlas, right? One gene, you have one piece of tissue. It's like the spatial transcriptomic stuff, but it's the grandmother of it. Okay, it was really Granny did not she couldn't do what you can do, right? Granny had to go slow, right? So it was one gene in one section, and but you could get the full spatial distribution on some level of where this gene was expressed. So what we did is that, what did we do with that? We did this thing we called the Anatomic Gene Expression Atlas, or AGEA. Just like we published a paper in this back 2007 or something like that. And so what we did was this: we took all those gene expression patterns and we did some image processing to segment out the cells, find the cells which were expressed. And now we formed averages of all those patterns, right? Spatial average. This is done by just by an embedding in a UMAP, but there we did, you know, we spatially averaged the patterns that we saw. So if you go look on the web, you can look up this AGEA, AGEA, Anatomic Gene Expression Atlas. You can see that what it was a pretty sort of a coarse representation of showing, using the in situ hybridization data, what would that tell you about the anatomic differences in the brain. It's a big thing. The neuroanatomists love this, right? Neuroanatomists, as we talked about yesterday, classically, they're interested in understanding, how does this subnucleus of thalamus really differ from this one? Is it really? And they look at histology things of like stains that only strain all neurons, and they see, oh, there's some kind of weird pattern here which is not there. That must be a different region. But now, with using the gene expression, you can look at that results and say, evidently, this region has some activity based on gene expression which differentiates from that. So you can build an atlas or a map of the regions of the brain completely driven by the gene expression. And we are close to doing that now using these MERFISH and these spatial transcriptomic methods. In the original AGEA, we had one gene at a time. So all we could do is take all the genes, do this averaging, come up with one map. But now with the MERFISH, we can take 500 genes at a time and really get fine structure to how the cell types are defining the brain. Okay. There was a question in the chat. Let me go get it. Oh, keeps going there. Can't go there. Doesn't drive from that. For that final pipeline with many different clustering, was any step method that didn't work in the beginning as you expected? Yeah, there's always steps. It's a imperfect work in progress. Is that package that I talked about? It's called HiCat, and that package it was developed by people in my group. Over many years, and it's evolved over many years, through different things. There are always steps along the way that, things that it didn't work out very well, decisions that had to be made. We tried to pick improved techniques. So I can't give you a specific example right now, but and I wouldn't want to say that this is the last word on clustering or anything like that. You can imagine always increasingly it was a functional thing that worked for us. Another thing too, I would say is this: it was all developed in R, but as we have increased now to the millions of cells that people are interested in, R became unsatisfactory for that. And so that whole package, we spent a big effort to rewrite the whole package in Python. So that's all available on a GitHub site. You can there's a tutorial for this HiCat thing should you want to learn to use that there. So. Okay. Yeah. Okay. Right. So good. Yeah. So every point in there, it's a good question. Every point in here, so these are the same, it's the same thing, as you can see, just colored differently, but every point in there is an individual cell. And that cell, it has a transcriptomic pattern. Let's say that in every cell, we looked at 5,000 genes in our after we got through things. Those were the high variable genes, the ones that we really cared about. So with every cell, there is associated a 5,000 vector, dimension vector, right? And now, of course, what the UMAP does, what this UMAP does, uniform manifold embedding, you've probably heard of it, right? What it's just doing is it's just doing this kind of nonlinear dimension collapsing to attempt to find a low dimensional representation that in some sense, approximates the actual distances in the origin between the vectors. So you're right. Remember in PCA or so PCA, if you take the reverse problem of PCA, rather, I'd like to find a representation that represents the best, mean square approximation that is principal. It would be the first K principal components would be the best K dimensional representation of that. The process for doing that is often referred to as what we call multidimensional scaling, right? MDS. All MDS is saying, look, I'm going to tell you the number of vectors I want for approximation, and I want your best, quadratic mean square loss function to that. Well, UMAP is doing that, but it's not respecting that optimality condition of mean square optimization. It's doing it in a highly nonlinear way based on, this was an improvement of the T stochastic neighbor embedding, the TSNA, which basically did this kind of, T distribution, sort of Gaussian-like nonlinear distance thing that said closer things should really be closer than the farther things. So it was a sort of nonlinear scaling, and the UMAP is just a somewhat more robust, statistically correct way of implementing that in some way, right? So all this is now is this, it's crazy in a way, right? This is part of the main problem of data analysis at this scale, right? Let's talk about that a little bit. What are the problems there? The problem is, and many people will know this. Lucas knows this. He's been living this day in and day out now. But the problem of this living in this high dimensional space versus what's really there and what you see. Nobody can do a data analysis. It's funny, when we talk about 10 dimensions, people don't even think of that as a high dimensional space, but it's ridiculous. It's already 10 dimensions. You're already, when you consider the random distribution, it's already way, way out there in the tails. You can't readily sample a 10-dimensional space too easily, but here we're working in very, very large dimensions, but there's this desire to project this down to something interpretable. And that's one of the problems with these methods, is that what appears to be close in these kind of things is visually satisfying, but is it actually really, close in some kind of transcriptomic sense? We need better methods for those kind of things. Today, biologists still use these, UMAP has largely replaced the TSNE because it's perceived to be more statistically valid, but it's not necessarily the curse of dimensionality is still applying to this just as well. You definitely get, if I were to, this is in this UMAP here, it's choosing a two-dimensional thing and saying, give me your best vision of what this data would look like as a two-dimensional approximation. 2 dimensions contracted down from 5,000 dimensions, okay? Well, we see coherence, but that doesn't, we cannot read into this, you can't read into this in any way that proximity really means proximity in the underlying space, okay? That's often a pitfall that we fall into. We need other ways of addressing that. And actually Lucas here works on, with Yusu, works on some of these methods of topological methods like Morse theory, where we can actually try to work in the higher dimensional space, but extract a lower dimensional manifold first before we do a dimension collapse. Very sophisticated methods, and still has problems, but it's a more maybe honest way to look at the problem of dimension reduction. So, but you're not going to change. People still want weight. They live what you have to live with, and actually people do this in 3D. Sometimes they'll do a 3-dimensional UMAP. You can pick any dimension you want for it. And the 3-dimensional will seem already to show what appears to be a group here when looked at it in 3D. As you know, you take a 2D object and you rotate it, you're starting, it actually bent like that, That's interesting, So I think that you have to be careful with these. and you can't read too much into it. It's just a visual, it's not a statement of biological coherency. It's more of a visualization of what you have. I think let's take a break for just a couple of minutes. And then we'll finish up. Let me just see here. I think I'm going to move on to the last section. You know, there's a lot of more information here about cell types, measuring distances, but I think you can best do you can review the slides and you can go into the tools. I want to tell you this. We built a whole bunch of Jupyter notebooks for messing around with this transcriptomic and spatial data. Okay, you can go to the website and you can see all these things. You can do these things. You can do these things yourself using the data through these Jupyter notebooks. Okay, they're Python notebooks, and they show you how to interact with this atlas to get spatial profiling, spatial distribution. Look at those colors. You can do those colors at home. Okay, so that's that. So anyway, let's call this. I'm going to leave this up here. I'm not even going to explain it. You can look at that and see if you can make any sense of it. But when we come back, we'll talk about the last segment. Okay. All right. Why don't we try to re-meet? Let's re-meet at 11:30.
What kind of spatial, or even combine this as epigenome data? If someone's working in that, what's a wish list, right? If I want to design a new way to organize the data, what would be the limitation of the current one? Yeah, what's your wish list? Is that here? I think you're always going to give a new method. You wanted to follow.
Well, how would you go about it? Kind of that to me. How would you go about doing it?
What should I improve? I don't even know, right?
Well, okay, so
I mean, there's
I'm
something
saying, yeah, it's interesting. So I'm going to tell you this, and it's still, you kind of see, I hope you see the basic idea, but you probably don't see, well, what could I do and what would be different and how would I approach this? Well, first of all, just like it is, we tend to, there's a grass is greener phenomenon. You meet somebody, you think they're doing something, and you think, well, God, what could I do? It's all quite basic in a way. In fact, there's all kinds of places where one could improve this, the actual determination of the clusters, the methods are done, are just, at the end of the day, basic iterative hierarchical kind of partitioning. And there's certainly opportunity for trying to envision other methods of grouping, any method you can think of for grouping data, and any other kinds of approaches. And as you know, there's 10 million clustering approaches, right?
But how do you know?
Right,
So the question is not so much what you can do. The question is,
I think
UMAP is you basically said that UMAP is better than P.
Right.
How
I didn't say it was better in some ultimate sense. What I said it was statistically more principled, meaning that the assumptions developing the algorithm were done slightly less ad hoc decisions. That's what I mean. It's more statistical.
So it's more like you're looking in the box. It's not like a measurement. You can't because I would suggest a measurement. I would basically say what you want is a mapping that preserves small distance. So if you can create a mapping into some representation that preserves, it doesn't need to be even uniform dimension, but it can be in some places 1 dimension, 2 dimension.
Oh, absolutely. Yeah.
But as long as you basically create a small representation, smaller than the original dimension, that it maintains local neighborhood.
But even that is not well defined, like, because then for long expense, do you allow them to put them to give it criteria? I can say one is that all the distance smaller than epsilon should be, still
Right.
something. And bigger than epsilon should be bigger than that. I don't care if it's small or not, I don't care, but it has to be bigger than strength. This is one, right? So I still start with small versus small. Or I can only say that I want to preserve small distance as much as possible. And then big one will give me 0.
So what do you think? So it seems like graphic clustering or diffusion methods. But what do they?
You mean what exactly they say about the distance?
I mean, they preserve small distance, but about long distances. It's not clear.
Yeah.
It's kind of like they try to flash.
Right, up to, but if you do this depend on kernel side, you know, exactly, bigger than a
Right,
certain
right.
place, really.
I think that the multi-resolution part is basically, you want to say, if I take groups according to these very fine, now I want the same kind of condition for all the three groups. Maybe that's.
Yeah, and I mean, but at least we're thinking this, the brain is, we know that there is this, the brain is, we know that gene expressing the brain and other things behave in this multi-resolution way, that there are only major differences in order to say, and then there are subtler differences when you approach the analysis. Right? You can't just put everything in there. You wouldn't see anything. Right? The noise in between. Right? The major thing or not all. Right? You have to do this multi-resolution. Oh, yeah, yeah, we met. Yeah, let me tell you how we met. Right, David. You might come down. Yeah, so you make a call and you go back. Not so the Society for Neuroscience meeting.
Oh, right.
Once in a while it's in San Diego.
Right.
Last year it was not this one, but it was the previous one.
Right.
You and David came down and we met.
Right.
Right. Yeah. Yeah, that's
especially now. I'm saying that so what I want to do now input that. It's not really what is the right it would be nice to have some innovation thing. Whatever needs fine.
There are many things you can do to be fast. Neuroinformatics, you can say, you know, I want to compare it to known neuronal affiliations, say that boundaries are defined different from them statistically, say gene regulation, show an association between not bound by, you know, a culture of yeah, the problem, you know, you were trying to optimize and that still is the problem. You know, I don't know. Exactly.
And so that's what we observe with big birds, because it's like very interactive. But I mean,
Yes, it was a like visualization. Right. But what you're saying is that for example, what you could do is kind of make a more equal way of doing analysis. That you can always do. Right. You go to the you have to set, yeah, visualize
Right.
Or you rely on them. Yes, yes. Slices of it.
That's right in every company. I
That's
just
right.
want to look at this cluster. I want to give more detail.
And then we do it with some of them. Yeah, so trying to look at, you know, the problem is we trying to look at what is the local variation in part of human expressing cell types, and how real are the clusters that were found here? We're using these methods to say how real were these distinctions, and how can you quantify the gradient of the trans connected?
I know.
If we're gonna laugh at her,
That's. I mean, no.
And then we'll also change the cell types.
Yeah.
Another thing to say is that, you are right. I don't think we've had any, this point is here, let's look at this group, right? This point is here and this point is here. This is, there's no evidence that this is in fact separate. It's a little farther away than that.
I see.
Like this.
Oh, I see. I see.
there's an introduction based on that. Assuming the uniform, there's a simple assumption, assuming that it's uniform cycle over the.
I see.
Nature of the manifold. Means it's very much somehow. Is it? I don't know, which one paper didn't. Well, this is.
There's still no.
It's
no
not
formal
really.
record.
There is one, there's the same.
Oh, no, what I mean is, I never saw a cleaned up.
Maybe
presentation.
not.
Yeah, never really, when they came out, it was everybody.
Sounds like it, I would say.
Oh, yeah, yeah.
Some of it, yeah,
Yeah.
it's really take off with
Oh, yeah.
the scientific.
It started, what started off was the TSNE was developed by, with Jeff Hinton,
Right.
with a student,
Right.
Martin's. And I knew this guy, he was from the University of Delft. Martin's, it was Martin's like PhD work. And, you know, it was a cool idea, everybody jumped on,
Right.
because it gave a way of, you know, doing some, doing something, getting close, having close, the closer is closer, did better than PCA, of showing the closeness.
Right.
No one will, I mean, PCA is more honest, but no one will show you a two-dimensional PCA plot.
Right.
You know, and so the UMass people realize that, okay, we see what you're driving at, but we can do this a little bit more sadistic principle, you know, and that's really what. Why don't we continue with that? Yeah.
Okay, we have a question here. Let's see, we have a question that's come in from our studio audience at home. Let's see. Could you please recommend a tool or method for analyzing RNA-seq data to compare gene expression levels between two specific cell types? As far as I understand, we've been through quite amount of clustering cell types, but I wonder how I can peak some genes that are more expressed in a specific cell type. Yeah. No, good question. So you in other words, you want to know, you know, you have, let's say you have gene expression from a set of cells, and so one thing you could do, I mean, so I talked about yesterday was that with that map my cells utility. I don't have a picture of it here, but you look up map my cells. This uses various either correlation or variational methods to actually take your data and find what are the closest cell types of those data. So if you had a set of RNA-seq data, you could use this method to map your cells to the types, and then you could try to compare the difference between how far those types were apart. The types would become a proxy for, you know, a kind of a, like an eigen direction in the cells that you had, and then you could compare those. In terms of actually just, you know, comparing expression levels between two groups, you know, that there's no specific way to do that other than, you know, just basic kind of, you know, statistical analysis if you want. But I guess what I would do is I would try to find the types associated with them. However, if your question is to answer how does the things vary between types themselves, there we have, you know, this kind of distance comes about in a most basic level from looking at where the two types are in the hierarchy, right? The hierarchy gives you some notion of how far apart they are. If they're close in the hierarchy, they are evidently closer in clustering. If they're farther apart, in other words, what's the merge length to get to a common cell type? You know, earliest common ancestor kind of thing would be some measure of distance. Oh, I hope that helps a little bit. Okay, welcome. Okay, so we're going to go on to the last segment here now. And let's see, the last segment. Again, there's a lot more information, and so we should figure out where we want to put the slides. Let's find yeah, try to find a, give me a site we can upload them to somewhere. Yeah. Okay. So here, the last way here, I'm going to talk about, here we're going to talk about some machine learning methods for integration of data. Oops. Okay, all righty. So integration of single cell data, part 5. So there's various approaches here that kind of thing. This has been work that's kind of done. The basic ideas here have come out of work of originally from Michael Jordan's group, Nir Joseph's group. Different people have contributed. These are kind of some of this approach started off with this paper from Lopez from Nir's lab called Deep Generative Modeling for Single Cell Transcriptomics. And then, so that what that was about is using, it was basically about just using kind of variational kind of inference to kind of find patterns in single cell transcriptomics and compare them. They also tried to compare them with epigenetic data too. And then another fellow, Auschalk, and a guy now who's in our group who came to the Allen Institute, Mariano Gabito, developed a kind of a method for kind of, combining modalities and generalizing this. We'll talk a little bit about that too. These are papers, they're in your reading material. We don't have time to go into all the details of this kind of thing, but you can look at hopefully I'll give you enough insight that you'll be able to look at these papers and read them now. And then there's another approach that came out of a different way of kind of combining epigenetic and transcriptomic from Bing Ren's grad, and Bing Ren is here at UCSD, and anyone maybe knows him or his group, but someone in his group, Dr. Zhang here, actually developed methods for doing that. So we'll talk about some of these here. Let's see what we got. So here's the paper here from Romain Lopez. This was the original paper here of deep generative modeling for single cell transcriptomics. So the idea is that, you have typical kind of thing of latent variables. In variational inference, what you're doing is you have latent variables. They're the variables that are you believe are driving the kind of structure that you see. You don't have access to these. You have sample data. You would like to sample from the posterior distribution of measuring these variables subject to the data that you see. But the problem is that this is a very difficult thing to measure. Classical way of doing this has often been kind of a Markov chain Monte Carlo, where you build up a kind of simulation pattern to simulate it. This posterior distribution, Gibbs sampling, these kind of things. We don't have to, this is not, we can't go into what all these things do too much without just have to direct you in the right direction. And in variational inference, but that's costly. This Gibbs is slow and costly to build up these kind of simulation techniques, approaches to sample this. They're very powerful, they're very accurate, and they're very good for lower dimensional problems. But if you have too much data, it becomes a bit of a problem. And so one of the innovations of, Michael Jordan and colleagues was this approach of taking this approach of approximating the posterior by essentially something that was a simpler family, and then looking how did this family differ from the actual difference using information theoretic distances, Kublik-Leiber distance, et cetera, things like that. So, again, the principles behind it here, the models typically assume a generalized linear model can be used to accurately map a low-dimensional manifold. But this SCVI, which was the package, the first paper we're talking about, developed by this Lopez, is a probabilistic approach for normalization and analysis of single-cell RNA-seq data using this kind of hierarchical Bayesian approach. And in which they're going to measure somehow the distance using kind of neural networks. And the latent representation is decoded by another transformation to generate this posterior and the parameters. So, and then oftentimes in gene expression, we often assume that gene expression values it turns out to be convenient use with this negative binomial distribution. People may have seen this kind of thing before. It has the advantage that it allows for more flexibility in the variance. Okay, oftentimes the problem of kind of a standard kind of something a little bit simpler distribution is that we don't have good control over actual variance as a kind of parameter. And this negative binomial distribution allows us both to co-vary the mean and the variance. This is kind of the model here. It's this hierarchical model of various assumptions of kind of, data that goes in. What do I want to say about this? Yeah. Maybe that's a little too technical maybe for now. Yeah, so this is a basic, let me see here. Yeah, this is probably the best example of it here, is that basically the sort of data that's coming in, there's a kind of neural networks which are doing almost like an autoencoder type of thing that is doing dimension reduction. We're measuring we use this to measure differences in means and in the latent space representation. And then we essentially, project back out to construct a posterior kind of distribution. So you can think of it as that we're using the neural network and this kind of autoencoder framework to essentially reproduce the final distribution here after the sampling. Let's see here, let's see. Yeah, I'm just getting kind of technical. I don't know what else to say. Let's see.
I guess the question is maybe what is the standard duration of?
Yeah, well, I think that this is one of the differences. So in a standard variation, you don't have to be, you don't necessarily use any kind of neural computation to do the dimension reduction. That could be an optimization, more of a standard optimization problem, right? I think that's what they're doing here as well. Yeah, yeah. I think it's this basic same approach. Yeah, I'm not super familiar with the details of this actually, so I have to. But using this, they're able to basically they can show that they get kind of a good signal. The signal is faithfully captured. They can separating. So they tried this here on sets of cells from the retina and the cortex and certain immunological data, and showed that these groups could be well separated. Let's see here. Do I have a? Yeah, going back to here, let's say we dissect this a little bit here more. Could not get into this. Yeah, so here the steps here says, given a gene expression matrix with annotations as input, this SCVI learns a nonlinear embedding of the cells that can be used for multiple analysis. In A, the neural networks are used to compute the embedding and the distribution of gene expression. Back here. Yeah, and then certain other kind of annotations that can be brought into the model here can be made available. The idea is that the annotations are things priors known about the cells and or their distribution. So there's a classical, then, you know, in this sort of hierarchical model, there's going to be this assumption that cells are coming from different classes. There's going to be a category, so there's an underlying categorical assumption of classes. Then there's a, you know, various means or log normal of the mean expression in each class. Then there's going to be the expression level, an expression level of cells in that class, which will be modeled by this negative binomial. And then we're using essentially the neural networks to kind of collapse this representation and get a simplified, you know, a simplified structure to this hierarchy. That's a basic idea. So in this, here's this generalization here. This was this generalization here where they were using it to incorporate a lot more kind of missing and other kinds of multiple data. This is this by this Ashwat and Gabito, this paper here. And so actually, so that SCVI, there are many versions of it were made. There's this first version, there's another, I put all these papers in your thing. Another version was applied to a protein kind of analysis. There was a version for epigenetic data. And so this method here, this multi-VI, is an effort to kind of merge some of these, right? So there's of this original method we just talked about, there was one for protiling gene expression, another one for chromatin accessibility, and then protein structure in the cell. And so this method here is a way of kind of trying to combine these methods. It's similar to what we talked about with those dueling kind of autoencoder type of things, where there's a individual SCVIs going, and then there's a penalty term between the two different methods and things like this. So yeah, here we see in this framework here, two deep neural networks termed encoders learn modality-specific, batch-corrected multivariate normal distributions that represent the latent state of the cell based on these observations from the expression and various metadata. And then to obtain the latent space, which reflects both modalities, we're using a penalization term between the two. How well do they match? Very similar to that approach we talked about earlier. And this ultimately gives us a kind of an integrated state, which is kind of an average between these two representations. And this kind of depicts it here. There's sort of a conceptual model here consisting, showing how to merge the chromatin accessibility and the transcriptome. And we have used this on our Alzheimer's data. We used it to do these kind of multiple measurements here, in which, so this is the framework, this framework of you know, dimension reduction using these autoencoders up here, and then coming from another method, and then there's a kind of a penalization and a merging phase here.
So it is assuming that you actually have, let's say, the same cell, but you know the two different modalities, or you actually have two independent, like one of the data is just for, say, the gene expression, the other is your
Yeah, so
FC, but you actually don't really have one-to-one response.
well, so that's good. That's a good question. So you do have part of it in this, and this is how you have it, is that on one hand you have let's say single cell RNA-seq data, you have single cell ATAC-seq data, but there's also, referring to this annotation kind of batch annotation thing, there's also data from the multiome, which was a certain subset in the same cell. And this provides a kind of a leverage or a linking on a subset, right? On some smaller subset. And so that's worked into the model. And that actually works to help with the penalty term between the two things. Yeah, so when we talk about batch corrected here, they're basically referring to, you know, different kind of vagaries that may happen because of differences in sampling protocols, differences between, you know, unavailability of data across the two different types. And again, this adversarial term penalizes if things are too distinct, very similar to what the Uyghur and his group did It's probably they were motivated by the same kind of things. This just shows it as a kind of a framework here. You're looking at in a low dimensional sense, you want this stratification into these subpopulations. You want visualization, et cetera. We want to remove kind of some of the unwanted variation. And then this high dimensional variation here representation, we're interested in the details and the kind of specifics of this. This just sort of shows the model here. This, again, latent space, a transcriptomical model, a model of chromatin, and all this can be built into standard kind of hierarchical framework, and it can be trained and sort of reduced in this way. And so we use this method here. This method was used here in this Alzheimer's data set to integrate, these different kind of RNA-seq and ATAC-seq of 2 million cells across these different varieties. Also, it was done to integrate 7 million cells across multiple brain regions. Okay, we can use this to kind of compare, and this was kind of it's a result that we present in the paper. This paper is available on bioRxiv. It's in review now. This is just showing an illustration of the method here. There are various approaches to this that this problem of data integration has been approached by others. A famous one is Seurat, right? That comes from Rahul Setia's group at the New York Genome Center. And their method is very different. Their method is more based on picking landmarks and matching landmarks, and uses kind of like a canonical correlation analysis to find, say what groups here are linearly related to what groups here. In other words, what are the drivers between two subsets in a linear space? In some sense, the multi-VI should be more powerful because it's using all this nonlinear neural network dimension reduction to kind of make the associations. And finally, let me just say finally that, using kind of similar type methods, Bing Ren and his group here have kind of produced this kind of big Python package for kind of, computing this sort of thing. This is a kind of a spectral type of approach, using Laplacians and graph embeddings here. But the key insight into what they're doing here is that, whereas it's very difficult and costly to compute kind of the modes in that, they use the well-known Lanczos algorithm to get the eigenvectors from the matrix without having to do, solve the full system. And so that kind of is a improvement in their technology there. Let's see here. Yeah, nonlinear. Let's look at some of this here. Nonlinear dimension reduction methods kind of address this thing through things like Laplacian-based algorithms, which are comparing them, but they're computationally challenging and inefficient with big memory usage. And so what they're doing here is that this innovation here is this kind of matrix without having to form the full matrix, this kind of getting at the spectral embedding, essentially by getting the eigenvectors through this Lanczos algorithm to derive them implicitly. And they're able to show that they can robustly get these. One of the problems with this original Lanczos algorithm was it in principle worked, but it was kind of numerically kind of unstable. It didn't really produce very kind of results that were often robust. But that has since then been improved by numerical analysts, people working in that field, quite a bit. So we had a question. Let's see what the question. A little bit off topic, but in the realm of RNA-seq, how would you distinguish between transcriptomic data that might correspond to changes in levels of downstream targets other than proteins, such as microRNAs? Yeah, well, I think that those things like this, you could try to work into the model. You could either, try to model the gene expression, which was connected with, microarrays directly, or you could try to deduce it from some of the analysis. I don't know, it's a pretty general, it's not off topic, but it's a pretty general question that, once you built your model, of course, you could use that to try to measure such predictions downstream. But, it's kind of sort of a pretty general idea. I don't really have too much more to say about that, I guess. Let's see. Yeah, so I want to mention a couple other things here. Yeah, this is this map my cells. So in the map my cells, we're using this kind of generative model here to assign cell type identity using the same principle which was in the SCVI. Okay, you have this target. The latent distribution are the actual cell types you'd like to find. You have the gene expression based thing. We use this encoder based method to find a best representation of this data, and you can try this. It's been benchmarked. We also have some kind of confidence score measurement that kind of tries to show you how well things are mapped in the data as well. Also, just saying a little bit about Seurat. I mean, that was kind of a thing that kind of predated some of the variational approaches, but it has been very powerful and used, and all those methods are available kind of online. Their approach again was using kind of it's type of a finding of kind of hubs and things of significance across data sets, and then using essentially landmark and basically kind of linear statistical approaches to match landmarks between visualized sets. It's actually very powerful, and it worked. And this was a kind of Seurat was used a lot in kind of single cell analysis in this thing. That's all available. Seurat was developed in R, and so you can actually easily install these libraries and run it to do some of this clustering yourself, and kind of show that. So Raoul built a first cell mapping kind of thing here, which is called Azimuth, which is based on Seurat. It really is just using the same principles of Seurat of having there's a target and a source kind of data set, and it's just using the principles of Seurat to do this mapping. We uploaded some of our atlases into this kind of framework, and you can do the mapping there too as well. But we believe in our map my cells now. We have an improved kind of a version of that. But I definitely encourage you to look at some of the Azimuth's kind of thing. Yeah. So finally, I want to mention this. This is the work of Jesse Gillis. We talked earlier about this meta neighbor idea. This was this idea of connecting two data sets, of looking essentially you're taking finding landmarks in the individual sets. You're looking at nearest neighbors and the environment of them in the specific data sets, and then you're doing kind of various graph matching techniques to figure which neighborhoods best correspond to which neighborhoods in the other one. This has been, people have used this a lot. We actually, so in one case here, we had these, so as part of the BICCN, the data, two big data generation efforts were kind of done. One, the Allen Institute did and also Evan McCosco's group from the Broad Institute did their own high profile, high resolution kind of approach here. This was all, again, this is another 10x type. These are both 10x type methods. It was actually, in fact, Evan was one of the pioneers in developing that method. And so here you have two different there's two papers. I think both those papers are in your reading materials, but both show the whole brain atlas as profiled by their two different techniques. And there's some, you can see that we worked with cells and they worked with nuclei, the nuclei of the cells, right? and you can see that, so there's a different number of clusters found. In one case, 5,100 some odd clusters. And using their method, they have 4,998 clusters, right? And so the question is, you want to know, is it, which of these are real and how do we align them and how do we validate that? What's the ground? So this is a good, again, we don't have biological ground truth, but we do have multiple data representing the same thing. And that is potentially a powerful way to make comparisons. And so what Jesse did, what was it? Jesse used this meta neighbor approach, where the individual nodes now are the cell types found in each thing. And then there's a notion of community kind of coalescing, where you look for what cell types are related to find the little networks here and the networks here. And then meta neighbor says, which of these neighbors match with which of these neighbors? And he does it in a kind of statistical way to find the best assumption. And when you do that, you know, he finds that, you know, there's good agreement down to some level, but that some of the clusters we probably need to kind of merge or throw away, right? That maybe, I don't think I have the number of surviving clusters on here, but I think it was something like in the order of more like 3,000 were reproducibly found between the two methods. Okay. And so that's that. So I think we're kind of out of time for now. I think, unfortunately, there's so much more we could talk about, and you can always write me if you want to know more, and we'll put the papers and all the slides in a good location.
Both they send, I think both is the image, and then they should have sent to everyone. But anyway, those will be.
Yeah, we'll put all this material somewhere, and in reading those papers, if something is unclear or you want to learn more, you can write me, and we'll try to clarify it. Also, you know, we didn't have too much time. Ideally, we would have liked to have some time to talk about projects, right? If people are interested, if you're interested in further projects, definitely let me know, because we can do that to you know, help to identify research directions. We can find things that you may have ways to apply some of the techniques that you know and learn to do things. I don't want to give you the impression that this is in any way you know, fait accompli. There's so much that can be done here. It's all, a lot of this analysis which have been done, I would call very preliminary. You know, much more can be done, and the people who have done it are no smarter than you, and so there's plenty to do. So thank you for your time.
Thanks. Well, I think that ends the Q&A tutorial, and let's Mike again, because it's actually a lot of time to prepare this tutorial that I'm all the way, and I think I really appreciate you coming. Thanks again, Mike.
Thank you. Thank you too.
We'll to the video later.
Yeah, thanks.